Confocal scanning laser microscopy. Now, confocal laser scanning microscopy is a technique that allows a researcher to focus a laser beam to a specific depth within a tissue to analyze the concentration of components at that level. This enables us to create concentration profiles for further research purposes. Now, this concept and this basic invention and this principle was first invented by Marvin Minsky. But this was totally went unnoticed because lack of intense light sources and computer power needed to handle such large data sets. Later on, as the computer advancement took place, the first laser scanning confocal microscope was developed in 1969 by David and Paul. Now, this is the principle. Now, to put it this in short, you can say uh, how this machine works is we first isolate the individual muscle cells and then we stain it and we put them on microscopic slide. And after that, now this is how this works. Now, once we did the previous steps, and put the slide and put the specimen on the microscopic slides. After that, these are the components of confocal microscope. Now this laser, this produces a light of a certain wavelength. This hits this dichroic mirror. Now what this whole setup and now this whole microscopy is different from various other microscopy is this confocal pinhole. Now, how this helps and this makes this whole microscopy different from the other microscopy, I'll explain that later. So first, let's come again to this. Now, this laser uh, produces a light source of certain wavelength that hits this dichroic mirror. Now, this dichroic mirror is basically a glass slab that is covered with thin metal coating. Now, this bounces off that light sorry now this bounces off that light from this objective lens to the specimen. Now this specimen again and another light source is produced back from this dichroic mirror and through this confocal pinhole to the detector. Now this detector gives us the imaging and the results. Now, even if any other light source is being passed to this dichroic mirror, can't be passed to this confocal pinhole. So this is what makes this microscopy different, and this is one of the advantage of this microscopy, as it gives us the control of depth of field and reduces the background information, and to get our results very accurate. Now, there are various applications of this microscopy. Now, the confocal microscope is used in a wide range of fields, including biomedical science, cell biology, genetics, microbiology, developmental biology, nanoscience, uh, quantum optics. In biomedical science, it is used in the analysis of eye corneal infections by quantifying and qualitatively analyzing the endothelial cells of the cornea, used to identify the presence of fungal elements in the corneal stroma. Also in pharmaceutical industries to ensure the maintenance of the thin film pharmaceuticals, allowing the control of the quality and uniformity of the drug distribution. It is used to retrieve data from some 3D optical storage system. This has helped in quantifying the age of Magellan's papyrus. Thank you.